So welcome everyone. For those who may be new to Greenwood programming, my name is Sarah Evans and I am the director of the Living Collection and curator here at Greenwood. And I have the privilege of supporting research related to the natural environment here. The Greenwood Historic Fund provides an annual fellowship to support urban ecological research, with Greenwood serving as a field site for these studies. Tonight, you're going to hear from the 2024 Research Fellows, who will share their projects and discuss the scientific questions driving their work, the data they collected and have analyzed so far, and the larger significance of their projects. Why does this matter? Why are we here? So first up is going to be Dr. Nina Bassuk, an emeritus professor from Cornell's Urban Horticulture Institute, who developed the initial propagation method for the hybrid oaks that she will be discussing. And following Dr. Bassuk will be Dr. Emily Herstoff and Dr. Michael Tessler. Both are biology professors at Brooklyn Universities. Emily teaches at St. Francis and Michael teaches at Medgar Evers. And they will be presenting their research where they did a comparative analysis of biodiversity amongst different species of the same genus. And we're going to learn the difference between species and genus. So with that, Dr. Bassuk, please take it away.
Okay, Sarah, thank you. Okay, there we go. Okay, well, I'm delighted to be here and to bring you into a 20-year project that's been going on at Cornell in the Urban Horticulture Institute. We have a team of people who you see there on your right, myself, Brian Dennick, Joan Gordario, Shannon Sweet, and Ryan Georgia. We've all been working on this project for many, many years. The idea is to develop and propagate better oaks, new oaks for the urban landscape. And the challenges of the urban landscape, you can see here a little snapshot of a sort of funny little protection for that tree, that orange snow fence, and all the destruction that happens to soils as we develop urban landscapes. But one of the things that we're most concerned about, or most interested to see, is the fact that we have before climate change became a word that we all know about, urban environments experience climate change in a big way. And here you can see this picture where on the right hand you have degrees Fahrenheit, and on what you're seeing is a park, and the color under that tree is basically 67 degrees. In the grass, you're at 181, and in the foreground, you're seeing sidewalk, which are 105 or so degrees. So it's the developed area, the sidewalks, the buildings, the roadways that actually increase the heat in the urban environment. And we see, okay, let's see if I can go forward with this. Oh, here we are. Excuse me, folks. So we see this kind of profile of heat in the urban environment because of all the built surfaces that radiate heat into the sky. And so that changes the environment for trees, especially in urban environment. So what we decided to do is to try to hybridize oaks, which oaks are tough trees to begin with, but thought we could do some work to actually make them more stress tolerant, both drought tolerant and alkaline soil tolerant. So that was one of our big things. We started with about 7 maternal parents. You can see, common bur oak and the chestnut oak and the swamp white oak and the chinkapin oak and the regal prince oak. These were our maternal parents. And then what we wanted to do to try to increase the stress and alkalinity soil tolerance is to cross these with 30 different pollen parents from basically western, droughty, alkaline soil places. Here are just a few of them. The Texas live oak, Quercus gambellia, gamble oak, which is from the Rocky Mountains, Quercus lyrata, which is an eastern plant, but tolerant of wet soil. So we wanted wet and dry, particularly dry, and alkaline tolerant. So it's just a show of a few of the pollen parents we used. And here's one of the big things we wanted to deal with was the chlorosis in urban areas. Here you see a pin oak in bright yellow in a upstate New York area where typically soils in urban areas are alkaline based on all the limestone, concrete, asphalt materials that raise the pH of the soil. And you can see the bright yellow one on the left. On the right, that tree has died now. It's not as if it just has nutrient deficiency, it actually eventually dies. So we wanted to tackle this issue by choosing plants that could tolerate, we can create plants that tolerated high alkaline soils. And so we choose, here's Quercus macrocarpa or bur oak. You can see the native range of this, a very wide native range, and some of it goes into Texas and Oklahoma, and the Great Plains, as well as into the east. And so we wanted to choose plants that could tolerate, could hybridize with some of our native species. Okay. And here's Gamble oak, which is a basically Rocky Mountain Western species. You can see the native range of this. And we actually, if you look at Texas and Oklahoma, there's actually a places where that overlaps. Gamble oak and macrocarpa overlap, and they can create a naturally occurring hybrid. So we actually use that as a maternal parent, and then crossed it with Lyrata, which is overcup oak, which has a southern wet species. And we got something pretty interesting. This is what we call fondly called 1819, which is Mac Gamble mother and a Lyrata or overcup oak father. And you get this very interesting leaf. We're very pleased about this tree. It actually looks great. It deals with high alkaline soils, and it has really good growth form. We also looked at, Sonoran desert oak, Quercus turbinella, and crossed that with our macrocarpa bur oak and got some interesting results there. Here, the macrocarpa. And this is the cross of macrocarpa and turbinella, so bur oak and Sonoran desert oak. And it really looks interesting. It's nothing like a bur oak leaf. It looks more like a turbinella or a Sonoran desert, almost like holly-like leaf. So we're getting all kinds of interesting forms and leaves. And of course, the next thing would be to test them to see how good they are in dealing with high alkaline soil. So that's the issue, is we were doing all these neat crosses, and this happened in 2004 to 2006. And then we had to propagate them clonally so that we could test them and challenge them with various different alkaline and droughty soil conditions. So here's what the method we used. We basically, if you cut, maybe people who propagated know, if you cut a tree down, the stumps that you get, the sprouts that you get are juvenile, non-flowering, and they are typically root better than those coming from the top part of the tree. So we use this technique to come up with a method of propagating. So if you start with the upper left corner, we covered these stumps after we cut them down with an opaque pot and let the new shoots grow out in the dark. The second, from that picture, you see the etiolated shoots growing from the stump. And those shoots would never live basically in full sun, so we had to wean them to light. But before we did that, we painted the bases of these etiolated shoots with rooting hormone, IBA, and these shoots are much more sensitive to rooting than would be a normal green shoot. We then wean them to light with an overturned trash bucket so that in a week's time they turn green, and we put a cone around them with potting mix, and about 5 months later we uncovered it and saw that we had roots at the base of those. So this is kind of a long process, but one that was pretty consistent. We got lots of nice shoots, rooted shoots. We took them off the original stump, and that stump lives to fight another day, and then we can cut it back and start again. So here are all our propagated oaks. We had about 30, let's say about 7 maternal parents and about 30 odd pollen parents. And so we had over 300 and so individual genotypes that we were trying to propagate so that we could test them for what we wanted them to be, both the alkaline tolerance and drought tolerance, as well as having decent form, disease resistance, other things we would expect for trees in the urban environment. So here's our tree that we would take the stumps, the rooted shoots off and pot them up. And here's one of the experiments we first did where we looked at we had these rooted shoots in pots, different pHs from 6 to 8, and looked at how chlorotic, how yellow the leaves were. We could test this by using a spad meter. A spad meter is a really neat tool that you can place on a leaf and get the greenness, how green that leaf is, which relates to chlorophyll in the leaf. And you see a low spad reading, which is that really yellow leaf on the top left, is less than 10. And going to the right, you have 10 to 20, getting a little bit greener. And then down left is 20 to 30, getting a little bit greener. And then what we would expect to see is a spad between 30 and 50, where you have a decent green leaf and it's going to be healthy and do what we expect it to. So we use the spad meter a lot to check for the greenness of the leaves. We also looked for plants that had a decent growth form. I mean, the one on the left is a fastigiate form of a macrocarpa open pollinated. The one on the right is a Muhlenbergia chinkapin oak times Prinoides, which has a terrible form. I mean, we'd never want to propagate that because it doesn't have a form that would be useful in the urban environment. So we looked at form, we looked at fall color. Here's a chinkapin oak times live oak, which has some neat fall color, which is not something we often see with oaks, but this would be a useful, desirable feature as well. And also pest and disease susceptibility is something we look for. On the left, you may think those are acorns, but they're actually galls. They're white oak galls, and they can be, really numerous and be detrimental to oak growth. On the right, you see powdery mildew, a disease that can be very common, especially when you have English oak parentage in there. So we were looking for disease resistance, good form, alkaline tolerance. And the method I showed you before, which is, we did all this laborious cutting back, covering it, painting it, and so on. And we did get rooted shoots, which is great, but we only got about 4 for each plant that we did. So that was not a very viable way of getting these into the nursery trade by creating large numbers. So we started with micropropagation, which we're still working on. But we're having some success. And here you see some new shoots, new apical shoots in a test tube. We put them into a nutrient agar, get them to grow, create callus there, as you see. And then we establish those shoots as they grow in the agar and then cut them, taking the leaves off to try to multiply them. And you can see on the very right side, those shoots that were cut from the original established shoot, again, growing and making more and more shoots. And then you basically go back, take those shoots, multiply them again, and you can get a continuous cycle of more and more plants. So much more efficient in terms of getting numbers, where the other method, although useful, was, 5 a year, which is not what we want. So this is what it looks like when you've got tissue culture oaks in agar and they're starting to grow and they're looking really good. And at some point if we don't want to continue multiplying them, we want to root them. So we do take them out of the agar, put them on a rooting medium, and voila, you have a little baby oak with roots on it, and we can plant that up and get it to grow. And also, more importantly, getting out into the trade, into the nursery industry, so it can be used in the urban environment, not just a fun project for Cornell. So just our hybrid oak project, which Greenwood was a big part of, we did a hybrid crosses in between 2004 and 2006. 7 maternal species, 36 pollen parents, 350 unique hybrid genotypes developed. And we planted these out over several years. And by the time they were getting big in the field, as you see here, and I said, well, we've got to get them out of here, or else we'll never get them out of here. So we offered communities all over New York State to get some of these trees. We would you see here, oh, here they are. We balled and burlapped them. We took them, and each community would get 5 trees if they wanted. They had to come and get them. And so we had 60 communities or so that originally got trees in New York State. Greenwood this was 2019. Greenwood, Joe Schrapp knew about this earlier. And so in 2018, he got 10 trees from this group, which are in Greenwood now. There they are going, they're being planted. So here, let's look at some of these hybrids that we're very excited about. Here's that Gamble macrocarpa, or Gamble oak times bur oak times overcup oak, which is one I showed you earlier. This is what it looks like planted in 2019 to 2024. So 5 years, really good growth, nice leaves. This is an alkaline soil, and it's doing very well in terms of its greenness. Same genotype in another location in Ithaca. And here's a cross that we're very excited about. We call this Waco, which basically is Warei times Cantonii. Warei is a cross within itself. It's a English oak times bur oak as one parent, and it's a like overcup oak times live oak as the pollen parent. So a four-way cross. And we have lots of clones of this particular cross and lots of genotypes. So the same kids, we have different acorns, and each acorn is different. I call it the same parent, different kids. And we have different forms, upright form, wide form. We are very excited about some of these. Here's one in Greenwood, which was one of the Wacos, basically an upright form that was planted in 2018. And you can see the leaf on that. And we look at this Tonawanda, the same genotype in the western part of New York, planted in 2019, so a little younger, but it's doing very well in that upright, narrow form. Another greenwood tree, which is a swamp white oak times Alliana. Alliana is a Japanese white oak, which is a really neat tree, getting the pollen from Arboreta in the United States. And we made this cross, and this particular tree is doing very, very well. This was a 2018 planting. And I just mentioned here, Joe, you can see me under the tree there trying to take the diameter of the stem. And Joe, just reminding you, if you could branch some of these trees up, it would be really helpful so I could measure the diameter of the stem.
Nina, we are in process of pruning, dormant pruning them
Good,
now.
because I was doing battle getting under these trees, and they were, doing battle back. So that would be great, because we're going to be doing a lot more measurements in the coming year.
Nina, real quick, there were a
Sure.
few latecomers to the start of your presentation. Can you just real quick review once again your goal of hybridizing oak species rather than planting straight species that are tolerant of alkaline soils?
Yeah, so there are some oaks that are tolerant of alkaline soils, but not that many in the eastern part of the United States. And so working with many different maternal parents, mostly eastern species, and crossing them with mostly western species to try to increase the alkalinity and tolerance and drought tolerance. So we're creating hybrids that can tolerate conditions that most of our eastern trees cannot. And that's the goal of the process. It's a long process, started in 2004, and here we are 2025. So it's a long process, and we're getting to the point where we really like some of these oaks. So I just showed before, I can go back. Yeah, that was the Greenwood Alliana swamp white oak times Japanese white oak, a different leaf form. And this is in Greenwood. And this one is in Fabius, New York, which is south of Syracuse. Basically the very similar form cross to the one we saw in Greenwood. And this is this really neat way of showing them all over the New York States. We go to visit these and take data on them, and then we can see out of these, oh, I don't know how many trees we planted, so many different genotypes. We're coming down to some of them we think are really worth the interest. They're consistent, they're beautiful, they're disease resistant, they're alkaline tolerant, and that's important. We need to get the alkaline tolerance in there because of urban environments being so alkaline, even if the soil, native soil, was not. So this is one that we think is quite a good one. And then the last one I'll just show you just for fun. This is one I really like. This is a macrocarpa bur oak, a particular strange times swamp chestnut oak, which is a southern species, not alkaline tolerant. Bur oak is alkaline tolerant, but you get this cross where it's very tall, very strong central leader, and this great leaf form, which is I just love it. So this is one of the one of our trees of interest that we hope to be able to propagate. So out of these hundreds and hundreds of trees, we want to choose the best ones, and we're still in the process of choosing those best ones that will be able to propagate, because the propagation process is difficult. It is genotype specific. We can get success with one genotype and not with the other. So we have to work out the recipe for almost every genotype we're interested in, and we have to limit the number we can do that with. So we're having some success, but we have a ways to go on that. Just a couple of data slides. Here is the SPAD. Remember, that's the rating of greenness of the leaf, the SPAD. And we want everything that has to be over 30. Some of the greenwood trees are in the high range. These are the trees of interest on the x-axis on the bottom. Different species or hybrids of interest. And most of them are in quite high, over 40. Even if they're over 35, that's perfectly good too. Some of the ones are over the 50s in greenwood, which are very, very dark green leaves. And if we compare that with those same greenwood trees with trees all over the state, the same genotypes, but all over the state, you see typically greenwood is a little bit higher, a little bit darker. The soil there is a little bit more acid, which might be part of it. But most of the other trees in alkaline soils are doing quite well too in the SPADs in the 40s and 50s. So got a long way to go, but we're happy that we have trees all over the state that we'll visit and take data on. This year. We're going to be taking much more data in terms of soils and leaves and tree height and so on, so we can get better choices of trees of interest that we continue to propagate. And I will thank you, Greenwood, for the fellowship. And if people want to know more about this, there's my website there, which you're happy to peruse all kinds of information there on oaks and other things. And I'll leave it with that.
Thank you, Nina. So we have a question coming in from Richard, who's asking if there's any information about how these hybrids support Lepidoptera species. So do you know if they're supporting any moths?
I haven't looked at that particularly. There's plenty of insects on these Lepidoptera on trees. It's not that if they're immune to this. The big ones we've seen are some of the oak galls, but I haven't measured the Lepidoptera.
Maybe for a future project. Yeah. And then I was hoping, could you go back to your last slide? Nope, maybe the one before that. The WA cross with the OP, what do you think is going on over there? That seems to be the only cross.
Oh, the one that's a warei, which is a swamp white oak times English oak, and with open pollination. So in that case, they were actually higher in other communities than they were in Greenwood. But we're splitting hairs here. If you're over 30, you're good. I mean,
Okay.
so we're splitting hairs here. So as I mentioned, I showed you the original chlorosis. If you're over 30, we're happy. If you're at 40 or 50, it's really dark green. And so people like that or they don't like that. Some people like a lighter color, but we're looking for the alkaline tolerance and the drought tolerance, which is something we'll be working on more.
That's great. So
Propagation. Yeah.
Everyone seems happy in these tough soils.
Yeah.
And Nancy, I think this question is directed to me. So we are monitoring our southern live oaks. We haven't measured the SPAD, so we haven't measured the chlorophyll in their leaves, but almost every single southern live oak that we have planted since 2015 has been thriving and growing pretty substantially. So they seem to be doing great. And then Charles is asking for hybrids with high SPAD numbers, was the soil pH measured at each tree?
Yes, it was. I wasn't going to throw a lot of data at you. We measured at each tree. We measured the pH of the soil, And Greenwood and a few eastern communities had a somewhat slightly acid soil, but only always the whole of the western part of New York, starting from the Hudson Valley to Buffalo, was alkaline soil. And more so because we were taking trees in urban sites, which had concrete and asphalt and all the alkalizing materials as well. Even though the parks around there or the woods might have been slightly acid, the urban environment is alkaline. And that's why it's quite amazing that all those western species, again, western from Hudson Valley all the way to Buffalo, had alkaline soil. We measured the pH of every soil of every tree's soil.
Thank you. Yeah, Greenwood soils were pretty lucky in that respect. That
5.8.
Yeah, exactly. All right. Last call for direct questions to Nina before we hand it over to Michael and Emily. Ooh. So AJ is asking, assuming you determine a successful hybrid species for urban conditions, what is the timeline for getting them into commercial nurseries?
Yeah, right. Well, we have about 150 micropropagated shoots that are rooted that we're going to send to nursery to be grown on. And so it's going to take a while. But again, we have let's say four genotypes that are looking promising in terms of micropropagation. It takes a long time, but that Gamble, Mac Gamble times Lyrata, the one I first showed you, that we have 110 rooted shoots that are going to a nursery to be trialed and grown on. And we have another one we have 50 shoots, and those are rooted shoots, and those are going on. But it takes a while to get each of these genotypes to respond to micropropagation. They don't all react the same, and that's, it's going to give us work for the next 20 years, I guess.
And then Michael is inquiring about what sort of the average pH range is for East Coast soils. I'm guessing that's city soils.
Well, it depends. You can have, I know the Greenwood soil is about 5.8, which is nicely slightly acid. But if you go out onto the streets and you have all kinds of alkaline concrete, asphalt, and so on, that soils will be higher. And typically when we look at soils in a really urban context, the soils pHs will be higher, anywhere from 7.8 to 8.2 alkaline, quite alkaline.
Mm-hmm. Oh, wow. All right. Thank you so much, Nina.
You're welcome.
We're gonna pass the screen over to Michael and Emily now.
All right. So yeah, so we're going to switch and talk about how things like galls are great for the world. Sorry, Nina, I know you said that they were killing all your plants.
Yeah.
So yeah, different side of the coin for our particular talk. Different types of problems, different types of questions. So I'm Michael Tessler. I'm a professor at CUNY at Medgar Evers College in Brooklyn, and my co-partner in crime, Emily Herstoff, is a professor at St. Francis College in Brooklyn. And the two of us, along with our PhD student, Liam Engel, who's at Fordham University, and Adana Smith, who's an undergraduate working with us at Medgar Evers College, along with a whole bunch of other students. But the four of us are going to be able to talk to you a little bit tonight about the work that we've been doing over the last several months. So what we're going to talk about in general is kind of the cost in terms of biodiversity associated with ornamental trees versus native trees, and the type of small-scale conservation that can be done using native trees, whether this is at Greenwood with its slightly acidic soils and beyond into other places. So to get started, I'm going to pass this over to Emily, who's going to talk about how we kind of got even to the starting point of thinking about working on trees, which is relatively new to us.
Yeah, so thank you, Michael. So I'm Emily Herstoff. So I'm at St. Francis College. And I'm just going to start by saying Michael and I both work in Brooklyn, and we're really curious about biodiversity, what lives here, what kind of interesting, cool things do we have to look at, plants and animals. And when I was first starting my job at St. Francis College, I initially focused on the East River. So the East River, if you've been to Brooklyn Bridge Park, is the water body that you see, and you have that great view of Manhattan across the water. And as an aquatics person, I was really interested in looking at zooplankton. So looking at things like jellyfish, which you're probably familiar with, baby lobsters, other things like copepods on that top right corner that Michael's circling there that you might be a little less familiar with, but they're super important. I wanted to go out with my students, get them seeing the cool stuff that exists in their own backyards in Brooklyn and starting to ask questions about all of the plants and animals that they saw around them. This was super fun. I got to take students outside. They were really curious and had lots of questions about the things that they were seeing and all of the diversity that was out there. And then we started to think kind of other projects that we could get our students involved with. And on the next slide, thank you, Michael, you'll see we started to pivot to doing some more work on land. This was pretty new to me, and working in these systems was a little bit new to Michael too. But we were really curious to start to explore biodiversity, looking in suburban meadows and fields, continuing to bring our students out to do work with us because lawns, these days, you might see some news articles where there's interest in trying to replace lawns with meadows as a conservation measure, with the thought being that meadows are much better for supporting wildlife. So we wanted to actually study whether meadows, even if they're small ones, can act to support biodiversity. We were doing this work in suburban New Jersey. So Michael's parents actually have a portion of their backyard that they've made into a meadow. And we were trying to think about these suburban meadows and lawns. And this was really cool, but a little bit of a hike for our students to get to. And then we realized that Greenwood Cemetery was in our backyards. Greenwood is an awesome place to take students. Most of our students are, from New York, or they're all living in New York now, but most of our students are native New Yorkers. And Greenwood Cemetery is their backyard. And Greenwood Cemetery has meadows that they've been working to establish, as well as more typical lawns that are mowed more often. So initially, when we started working in Greenwood, we were exploring some of the same stuff that we've been looking at in those suburban meadows and yards work, trying to look at biodiversity with all of the things living in the soils. And we wanted to kind of explore these microbial communities, look at the chemistry of these microbial communities, look at some of these amazing meadow habitats like you see at the Hill of Graves in this photo by Greenwood, and then try to explore that. But as we were walking, we noticed some really interesting stuff. Greenwood is amazing because it's an arboretum. So there are tons and tons of really beautiful trees, really big trees. And on the backside of the Hill of Graves, which is one of our main field areas that we were doing this soil work, we were looking at the linden trees, and we noticed these really funny little bumps on the leaves. These bumps are called galls. And those galls that you can see there, those are red nail mite galls. So they look like little red fingernails. And they're on the Native American linden and the basswood, and they're also on the European linden as well. These funny little structures are made by insects. So insects are going to be going into the leaf, and they have this interaction with the leaf that will cause it to make this funny looking structure. And they're pretty obvious. Some of them can be really wacky looking. And we started to get really curious because these trees, the European and the American lindens, are really close cousins. So scientifically, that means they're in the same genus. So they're in the same kind of right below above species level group, but they're different species. So they're both Tilia, but one is Tilia cordata, the European, and one is Tilia americana, the American basswood. So we thought this might be a really great study system to work with our students in because galls are super important. They are not only an indication of being a place for those insects that make the galls to live, but galls can be important for a variety of other reasons as well. things like birds can eat galls, and galls can even have a lot to do with helping trees divert carbon to their roots and actually help fight climate change, which is pretty cool. So that's kind of why we ended up in Greenwood. But I'm going to ping it over to Michael now, who's going to tell you a little bit more about trees and kind of what we started to really think about in Greenwood Cemetery.
So yeah, before I hop to the next slide, this we like this picture because it's a nice comparison of the galls that we were finding right here. And you know, as biology professors, we're always trying to get things that our students can get their hands on and count. These look very different as leaves. They often look very similar as leaves. They're pretty closely related plants. Sometimes they're obviously different like this. The European can be a little smaller and a little darker. The American can be a little more offset at the base. But they often look really, really similar depending on the conditions that they're growing in. And that's because they're fairly closely related, right? They're in the same genus. So we would call these lindens or basswoods. You might think of another group like what Nina was talking about, the oaks, right? So they're all reasonably close to each other. They all kind of look similar. If you remember from Nina's talk, lots of pictures of leaves that look kind of like these blurry oak leaves in the background, right? Better pictures than Nina's of those actual leaves. But anyway, so for these groups of trees, we've got this pretty cool setup in Greenwood where there are lots and lots of species of trees, many of them planted multiple times, many of them closely related. And so we got interested in, you know, kind of that European, you know, the non-native versus the Native American ones, and seeing, well, what's the difference going on with these in terms of the biodiversity that they support. And the reason we're particularly interested is because these are common trees in the Northeast. They are planted, whether they are the native or the non-native species, they are planted commonly throughout parks, throughout a variety of different places within our local areas, right? Along streets, those obviously have different challenges, like Nina mentioned, with things like really high pHs for that. But you know, and then kind of, does this matter? Does it matter if you have, let me see if I can go back, or right here is fine. Does it matter? Is it a good thing if, now I can't get my cursor. Is it a good thing if you have a leaf like this, right? Where you have this really large gall right here and here and a smaller gall here. These are two different species of gall. These ones formed by wasps. I'm not sure, but probably wasps for these. Most of the ones on oaks, I believe, are wasps. So as well as these holes on leaves, right? These are things that are actively eating the leaves. So what we wanted to do was compare the natives and non-natives and see, do they have more of the galls and the bite marks out of them for the system that we're looking at? And kind of, again, from a little bit of a different viewpoint, as these support native ecosystems in terms of the biodiversity that they host, our kind of go-to is that the more things that are hosted, the better. And oaks are known to host a lot of different things. I believe there was a question about Lepidoptera, which are things like butterflies and moths. Oaks seem to be pretty exceptional at hosting things like those butterflies and moths. So what we were wondering for our arthropods, our insects and mites, do we get more interactions either through these bites, through herbivory, or through gall marks? Do we get more of those on the natives? And again, rather than us thinking of that as a bad thing, for us as kind of biodiversity inclined scientists, we think of that as actually being a good thing. And again, we could compare within a single genus. So this is kind of a comparison chart of what we were working on. I've lost my mouse again. Okay, I don't see where my mouse went. But anyway, you can see on the kind of top left of those leaves, those are maple leaves. So Acer rubrum, this is the red maple. And then we have the Norway maple next to it. The light blues are our natives. The darker blues are our non-natives in the pairs. And being able to pair really helped to compare, is this kind of the native difference that's causing this, or is it something else? So some other studies in the past have compared native versus typically invasive species, but usually those invasive species are pretty distantly related. and very different in terms of a lot of their function. Whereas things like Norway maple is very, very similar to a red maple in terms of where it can grow, how it grows, its growth form, its size, things like that. So we have the maples up top, we have the lindens or basswoods, and then the oaks on the bottom. We also did several coniferous species, the pines and the spruces, although we are comparing other data on them. We did not find much in terms of herbivory and galling. So the map on the left shows our kind of sampling design, pairing up these species spread throughout. We kind of randomly selected from the individuals that were available, places or trees to sample. So Greenwood is a pretty big area, and so this involved us walking a lot on hot days and having Liam and Adana walk even more on hot days. So with that, I will pass this over to Liam, who's going to walk us through some of the results that we have.
Hi, everyone. Yeah, I'm Liam. I'm a PhD student that's been working with Michael and Emily on this project. So first, for our herbivory study, again, we were looking at the amount of bites out of both the native and non-native leaves for specifically the deciduous or broadleaf trees, as seen in these images in the middle of an oak on the top and a linden in the middle and a maple on the bottom. So to do the survey, we first randomly selected 10 leaves from each tree we went to, and then we took a photo of each leaf. And on the left, you can see an image of Adana and another undergraduate student, Dunya, walking up to one of our lindens. And then I did an online herbivory trainer where I was kind of rapid fire identifying the percentage of herbivory on each leaf, which was pretty fun and silly. So you can see a screenshot of what that looked like on the left bottom. And you can also try it for yourself. I'll put a link in the chat at the end of this because it's kind of fun and a random skill you could have. So then after averaging the amount of herbivory for the leaves of the trees, we came up with the plot you see on the right. So that's comparing the herbivory on each genus, with the light blue bar showing the native trees and the dark blue bar showing the non-natives. And overall, our native trees typically had either the same or more herbivory than their non-native pairs. So for the oaks at the top and the maples at the bottom, the native species had more herbivory, which was statistically different. And for the lindens in the middle, the native basswood had about the same herbivory as the non-native little leaf linden, which is a European species. So this tells us that herbivores, which primarily insects in this case, seem to be preferentially eating the native leaves for some types of trees, while for the lindens, insects don't seem to be differentiating between the natives and the non-native species. So then going to our survey of galls, we, in this case, were counting the number of galls and identifying the species of galls that we found on our study trees. So to accomplish this, we did a 4-minute survey around each tree where we looked for any galls and then took pictures of all the ones that we found for later identification. And one example on a white oak leaf, Quercus alba, is shown here. And some of the variety we found is shown on the next page, which I'll get to shortly. So generally, the native tree species had a greater abundance and a much greater species richness of galls than our non-natives. So 16 species of galls were found on the natives versus only 1 on the non-natives. And these galls came from a variety of arthropods, including wasps, mites, and midges. And you can see this pattern clearly for our oaks and lindens on the right. So our native oaks accounted for the most types of galls we found, primarily formed by a variety of wasp species, like Michael mentioned, which is true for this one in the image, Acraspis pesimacoides as well. And the native basswood linden also had a greater abundance of galls. But for this genus, we only saw 1 species of gall the whole time, and it was also found on the non-native. So that species seems to be using both trees in a similar manner. And then for our maples at the bottom, we only saw 1 gall species at all the whole time on a single native tree. So that is shown as like the outlier with the arrow pointing at it on the graph.
Hey, Liam, sorry,
yeah.
can I
Go ahead.
interject? because I would like you three as a group to go through why the, insects would be feeding, on the leaves, but also why they would be, what's the hypothesis is why they are feeding on the trees that are indigenous to this area.
Yeah, I do think I'm Michael, Emily, feel free to interject. I do think we're gonna we'll get to some of this significance of why we think a little bit, but I can Sarah, if you'd rather me go through it now, I can too.
So I think maybe the significance, but and Liam, I'll jump in for a sec. Basically, plants make a lot of different compounds. So these are, if you think about things that we consume from plants, that might be something like caffeine that has a really bitter taste and makes you jittery, right? It makes us wake up and we love it. But if you had that by accident and you were, surprised, you would really hate it. You'd bite it and probably spit it out, right? So plants make a huge number of compounds. They're highly, highly diverse. And there is a strong chance that species have been engaging with them and trying to eat them for millions of years. Because plants can't run away, they make these secondary compounds to defend themselves, right? So most of the plants that we eat when we eat their leaves, these are very fast growing leaves, and their strategy is more grow fast, kind of live fast, die young, rather than something like an oak, which is really grow slow, live long. And their goal is for you to not really consume all of them. I've never taken a bite of an oak leaf, but I imagine it tastes pretty nasty. And that would be true for most leaves if you walked around. So oftentimes insects are pretty specialized on these things in terms of what they can eat. I'm not 100% sure how well studied that phenomenon is. In terms of native versus non-natives. It is to some degree, though. But in large part, it's that species have been evolving for millions of years together, and that they have, kind of have this arms race against each other, where one species tries to eat, the other species tries to defend, and they keep building up a tolerance and keep building up, more of these background.
And because we have moved plants around, because we're taking these trees from Europe, for example, over to North America, you might expect, therefore, that a tree that's not from this area, not original to this area, insects might not be able to tolerate all those nasty chemicals that Michael was talking about, perhaps because they don't have that long history evolving with that species that they like they would with a native species, for example. So we think that perhaps there might be this mismatch where we are taking over new things like the Norway maple or the little leaf linden, and those might not have pests. From our point of view, they're great, but pests on them in the same way that some of the native species might have. So that's kind of the logic behind this.
Yeah.
So,
just another. Oh, sorry, Sarah.
Oh yeah, I think if I were to synthesize what you all are getting at, I would say that the insects that are indigenous to this area, right, that live here, they have the flavor for the species that have evolved over time here. And instead of seeing them as pests, we can see as these are the trees that are serve as nurseries to these growing and developing insects. Am I right?
Yeah, 100%.
All right.
The first
Just
time
wanted
you try a food that's really foreign to you, you might not like it. But if you try it, over thousands of years, maybe it becomes your favorite thing.
Yes.
And most of these species have been here for hundreds of years, several hundred, but not thousands or millions. And for insects, it might take a long time for them to really get into those flavors. So I think we'll pass it back to Liam. I think you were about to go on to this slide. Is that right?
Yeah, perfect. Yeah, and thank you for explaining that so well, everyone, and synthesizing Sarah. Which I'll get to in a second as well, but it's a good background for what we're even looking at. So yeah, the last thing I was going to say about these galls is that since we found such clear distinctions between the natives and non-natives, which again is due to those natives being utilized basically as nurseries, like Sarah said, for the arthropods. We explored oaks in particular a little further with another species pairing, again at Greenwood. So for these, we are looking at the native pin oak, which is what you're seeing leaves of here, and also the non-native sawtooth oak. And we found a very similar pattern as the other galling, as the other trees that we looked for galling at. And so these are all pin oaks because the non-native sawtooth oak, like many of the other non-natives, had no galls at all. So this is just showing an example of like the diversity of galls that we saw for just one tree species. And these are likely all wasps, mostly in this case, that have like co-evolved with this particular pin oak or like only very similar oaks for, again, like, thousands, at least hundreds of thousands of years, millions, in order to develop this relationship. So because of finding that pattern once again, we went one step further and looked at one other different site in Brooklyn, which was the Brooklyn Botanic Garden. So I then surveyed galls on both native and non-native oak species in particular at Brooklyn Botanic Garden, which is very close to Greenwood, with just Prospect Park in between, basically. And again, we found very similar results with a variety of different gall species on the 4 native oaks I looked at, and all the non-natives I looked at, which there was 8 different oak species there that like had enough samples for me to kind of have a proper look at. They did not have any single gall species on them. So like you guys kind of got to, we think that this pattern of galling species utilizing native tree species is likely due to like long-standing evolutionary relationships that the insects have with the native trees. And even though these non-native trees, since they're within the same genus in a lot of cases as the natives, they have lots of similarities with the natives. The local insects are just not utilizing them in the same way. So to explain this in a different way, Emily is going to talk about some of the leaf chemistry of these trees.
All right. So like we were saying, one of the things about plants is they can't run away, so they make a lot of variety of chemical compounds. Besides those chemical compounds that they're making, well, all of those chemical compounds that they're making, and even the tissue that they're building themselves out of are made of elements. So things like carbon, nitrogen, phosphorus, many other elements as well. And you can actually look at a picture of a tree's chemistry and try to understand, is there a difference? Perhaps all of the differences we were seeing that Liam was describing about native species having more herbivory and having more galling than the non-native species, well, you might say to yourself, that's only just because the chemistry is different. Maybe the chemistry is really different between the native and the non-natives, so of course the natives get picked. So we decided to just check this. So what we did was we went out and we sampled the leaves from all of these trees in the cemetery to analyze the chemistry of those leaves and see, did they differ? So what you're seeing here, all of the colors represent the different genus, all the different genre that we're looking at for these trees. So you have Acer, which is your maple, Quercus, which is your blue, is your oak, and Tilia, which is your linden. So those are the ones that you were seeing on the previous slides that Liam was showing you. Remember, we didn't really focus on the Pinus and the Picea, the spruce and the conifers. Those are in the green. Because we just didn't see herbivory on them, we didn't really see galling on them, so we just didn't talk about them on those slides because we weren't seeing anything. But when we look at the chemistry of these leaves, you can see a few big things. Oh, and I should also mention too, my apologies, the shape of the dot indicates the kind of tree, with the circles being your native trees and the triangles being your non-native trees. The figure that you're seeing here is called a PCA analysis. It doesn't really matter what it's showing you, but the big takeaway are the patterns that you can see visually. First, you can see the red, the blue, and the purple. All of those deciduous trees kind of fall in one clump, and the conifers fall in a second clump over on the other side of the graph. So you can see a strong difference in the chemistry of the leaves depending on whether it's a deciduous tree like an oak or an evergreen tree like a spruce. The second big thing that you can see is that the dots and the triangles are all kind of sitting on top of each other. So if I go ahead and pick on my quercus, which is the blue, you can see that the native circles and the non-native triangles, so the Q alba, the native, and the Q robur, the non-native, those leaves are on the other side of the graph there, they're all sitting together on top of each other. So chemically, these leaves are not distinct from each other. A quercus, an oak leaf, might be distinct from a tilia leaf, but native versus non-native doesn't seem to have a strong chemical signal. That's important because it means, at least chemically, for our insects, our gallers, our insects chewing all those holes, maybe we're not seeing a strong chemical reason why there should be a difference on those trees, but we do see that difference on those trees, which means that there's some pretty important conservation implications of all of this study as a whole. So I'm going to ping it over to Michael once again.
So yeah, so a lot of what Emily was just going through was kind of a scientific control, kind of making sure that it wasn't something unexpected like the baseline chemistry of all of these plants. And it doesn't seem to be that that's the case. So pretty much what this says is that in our much more controlled study than people have done in the past, natives really make a big difference in terms of arthropods, in terms of the insects, in terms of the mites, in terms of these galls, in terms of herbivory. We really up these things, which plant growers aren't necessarily all that interested in. But again, if you think about it a little more like Sarah, if the goal of a tree is to be a nursery for all the organisms within a given area, this is really exciting news that if you choose one species or another, you can make a big difference to how many different species you are able to harbor. And it's important to note that, again, a lot of the European or Asian species that we were looking at have been here for many hundreds of years, pretty early on in the history of the country. And yet they still don't have these relationships established. Like the English oak, the non-native oak, has been here for several hundred years, and yet it has no galls that we found within our study site. And yet all the natives that we found had many, many galls for oaks. And again, that was repeated with another oak species, the pin oak, and then again at Brooklyn Botanic Garden. So we're pretty excited about this study, but there is kind of one other aspect, which is the educational aspect, that we're always looking to foster the next generation of scientists. And so we had a lot of different students working on this project. These were 4 of the students who worked most closely with us. This is several of our students, and the 2 right here. So this is Liam and Adana. You've heard from Liam. And Adana, who is an undergrad, is going to just talk a little bit about the experience this summer briefly, and then Liam's going to wrap up, and that's what we've got for you. So yeah, Adana.
Hi, I'm Adana. So, I'm very excited to be on this project, and a typical summer day, at Greenwood Cemetery for me were amazing. I would say, I had a lot of fun. I learned a lot. I had the opportunity to work with Liam and other students. On hot days, on average, we were out there in the field like 7 to 8 hours. I learned how to collect samples, which was a great experience. I had the opportunity to be out in nature. I surrounded by the trees, hearing the wind, the leaves. It just reminds me, it just takes me back to my home, my homeland, Trinidad. It was an amazing experience. Liam?
Yeah, just to reiterate, like what Adon was saying, we had so much fun in Greenwood, even on the really hot days, which seemed to be just about every day we had planned for field work somehow, but we did manage. And I also wanted to say that, like, there's members of my lab that are working in areas all over the world, like Madagascar and Colombia and the Sahara, but I've always been really interested in doing local urban research that is helping to explain the environment that all of us encounter on a really day-to-day basis. So being able to perform fieldwork with Adana and the other undergraduates at Greenwood was a perfect opportunity to realize that. And it also provided very valuable research results that are aiding our understanding of what trees should really be planted in urban areas if we want to promote biodiversity. And as a final note, we just submitted our first manuscript for publication, which was on our leaf chemistry results. And we will be submitting our results for publication about the galling and herbivory, which will be my first first author manuscript for my PhD research. We should be submitting that next week or so. So I just want to say thank you for listening to all of us, and thank you to Greenwood for supporting our work.
Thank you, Adana, Liam, Emily, and Michael. I have like 8 questions, so I'm gonna remind the audience to submit their own. Jill just asked if there were no significant differences in chemistry between the native and non-native trees. Does that invalidate the theory that herbivores prefer native trees because they've adapted to native trees' chemical defenses?
That's a really awesome question. So thank you very much, Jill, for asking. So one of the things that, so the chemistry that we were looking at in the leaves was, well, elemental content. So how much carbon, how much nitrogen, how much magnesium, for example, all of these elements that you see on the periodic table, how much of those are in the leaves when you do the analysis? One of the things we didn't look at is defense compounds. So how many tannins do you make? So tannins are a common compound in leaves that might make them tough to eat or nasty to eat or taste bad. So while we didn't test that, that would be a really interesting next step to test the difference in things like tannin content between natives and non-natives. But based on just the simple elemental chemistry, there's no strong difference. So even if we're not seeing that elemental difference, it doesn't mean that there's not some secondary compound that could be really important as a defense against herbivores that we just haven't gotten around to looking at yet. Some other people have studied those defensive compounds in native and non-native things, but they're usually looking at invasive plants, and those invasive plants are not really closely related to the native plants. So those are complicating layers that we were able to cut down by using the amazing pairing in the Arboretum that is Greenwood Cemetery to study plants and looking at differences on how they're used and also their chemistry. I hope that answered your question. Please let me know. Awesome. Cool. Thank you very much. Excellent, excellent question.
Yeah. Molly is asking, is anyone making plans to compare galling and herbivory on those Cornell hybrids with Greenwood's indigenous oak species? And I think the answer is maybe. We must.
Yeah, we don't know the answer to that, right? And
Yeah.
a lot of it takes time for these trees to grow up before you can see it. My hunch would be that these would be in the hybrids would be in the middle. So those are hybrids of at least US species, but many of those hybrids are actually pretty local. And so I'm sure there are some. I'm sure Nina could talk more about that.
And then, oh, is Nina back? And then can we assume, or were you able to identify all the gall species? And are these species indigenous to this area?
Yeah, I can take this one. So we were able to identify all of the gall species. And I have to post another link too. But I can post a link to my iNaturalist because I put them all up on there, and those are being identified just by community members as well as some local experts. So we do know those species, and that they are local, and that I think that would lead you to expect that if the European plants were in their native habitat, that you might see galling there that you're not seeing here.
There is an expert, Matthew. What's Matthew's last name?
Wills, yes.
Yeah, so there's a big iNaturalist community. A lot of people go out and take photos of things, and one of the best within our local area, Matthew, has been the best. Yeah, definitely the best. Has been taking lots of pictures of galls, and so there's a really well-established baseline for us to work with, and that's true throughout New York City, even in other places. There's some good websites like gall-formers that you can get out there, but there are some interesting ones. So the tilia, the only one where you get, so the linden or basswood, the only one where you get the species on both the native and the non-native, we don't actually know where it comes from. There are guesses out there. I've tried to think about whether I could figure it out by going back to several hundred-year-old herbarium specimens, but it's surprisingly hard to figure out. But for the most part, they're all native gall species. But that's a great question.
Okay. And then Lauren is actually getting to just like an issue that I really, one of the reasons why I love both of these projects is because we are city residents. We live in an urban environment. As Nina was explaining earlier, cities have been experiencing just, they experience more climate change first before the rural areas. We're sort of like leading the way and experiencing hotter heat waves and more flooding and everything else, just the way that our cities are designed and built. And so with this comes pretty harsh challenges for all of our plant friends and our wildlife friends. And so Lauren is asking or stating that the traditional view is that insects are bad for trees. And she's asking, do all these insects, are they reducing their strength? Do the native species that support more insect species, are they as healthy as their non-native relatives? And he answered, why don't you answer first?
So yeah, it's a great question. And to some degree, the non-natives are planted because anecdotally, they get less bites and galls and things that people don't necessarily want to see. Although I will point out that some of them, like this hedgehog gall, at least in my opinion, are pretty beautiful and cute and fuzzy and you can pet them. But anyway, the fact of the matter is that's not always well studied. If you look at much of the galling literature, it is that these do not have a significant negative. Although I'm sure, again, Nina can, as a plant grower, can talk about that from a different light. And it does seem like, some of the galling issues for some of those hybrids could have been real and significant. What I can tell you is that we looked at many, many species, and you can see many of these species out in the wild. If you go take a walk in the woods, you will find galls on them. The standard natural order of things is that species are parasitized. So usually the largest, healthiest species, whether that's a tree or a tuna fish, they're covered in parasites. They have lots and lots of parasites, and they host they're nurseries for many, many things. And so this is true for plants. It's true for animals. So sometimes if something's in a weakened state, these things can have a big negative impact. But if they have good growing conditions, usually it's thought that these galls do not have a negative downside other than aesthetics if you don't like them.
And while some people might think they look gross and creepy, like Michael was saying, they're super important for supporting a lot of biodiversity. And on trees, not only are these galls important for the insects that are making them, but they're also important food for things like birds. So having these galls present is actually, helping Greenwood support all of the bird diversity that we have, that we see out in the cemetery as well. So there are lots of positive effects of having galls. Even if you look at them and you might think they're pretty yucky and gross at first, I, like Michael, would argue that mini galls are very cool looking, and things like these hedgehog galls are adorable and fuzzy. So I think they're really wonderful. But if a tree is healthy, it shouldn't be a problem. If we're looking at more stressed trees in the future, maybe it starts to become a problem, but it's probably a problem for more than just native trees versus non-native trees. Trees, like Nina was saying, might just be becoming more stressed because it's hotter, because the soil chemistry is changing, because it's drier. And all of those are major stressors that are going to be, I think, more of a problem to a tree than having a bunch of galls.
And most of the time, the trees aren't riddled with galls. Oftentimes, you have to look very hard for them. There are a couple cases of oaks that we saw where there certainly have heavy amounts of galling. And the Tilias, the linden basswood group, those pretty consistently have galls. If you walk all around the city, you'll see, flip over a leaf, they almost always have them, at least for some of the leaves. And those trees are doing great. They're some of our happiest, healthiest urban plants. They've been growing for a long time and they do pretty well. But yeah, it's tricky, and plant growers versus biodiversity environmentalists sometimes have different opinions about these things.
Yeah, well, I think this actually gets to the question that Molly just dropped in, which was, what does Greenwood want from its trees? Is there a hierarchy of desired traits? If so, when does support of the local fauna fall in within that hierarchy? And I think people can raise their hands, but I might be, I don't know, I'm not, because I know Richard is on this call. As, landscape stewards, land stewards, especially of a institution that is public and made available to the public, there are, and embedded within just like the complexities and challenges that are just happening, whether it is insect or pathogen pressure that is surmounting because of our extended growing season, our hotter temperatures, our droughty conditions matched up with like extreme wet conditions, those different changes, introductions of new pathogens that via the nursery trade. That's a whole other discussion. I don't necessarily see it as a hierarchy. I know that when I came into, when I first started at Greenwood, the state of our tree collection, we are an arboretum, right? So all of our trees are accessioned and they're mapped, and the data is made available to you guys. It is being managed more as a collection, and coming on as a collection, it was a pretty vulnerable collection, right? Over 10% of our trees were maples, specifically of the Acer genus, and like 95% of the maples were just Norway maples. Something that we're seeing with our Norway maples is that, even though they propagate and spread really quickly, they're actually not doing so hot in this changing climate. And so we are, Molly, to answer your question, is we are trying to really balance a lot of different things. And it's sort of like right plant, right place. So we are preferentially reintroducing reforesting with a lot of the species of the genre that are more indigenous to our ecotype, right? So not just indigenous to New York, but have like a wide indigenous range in North America and on the East Coast, while also prioritizing, like really understanding sort of the environmental stressors that exist for the plants here. So we do want to introduce like the Cornell hybrid oaks, amazing tool in the toolbox to be able to like have trees that can withstand and prosper in really challenging soil conditions that are actually really hard to change. It's kind of like a feral effect of just like our construction materials and how we build out the city. So yeah, there's no hierarchy. Our goals is to support wildlife, support local fauna, while also stewarding and growing a really resilient collection of trees. And so inherently that means a more diverse collection of trees as well. I hope that was that long-winded answer answered that question. And then Charles.
Wait, can Sarah, let me.
Oh, yes. Hi, Joanne.
I just want to say just a couple little things.
Yes. Joanne, can you introduce yourself, please?
Oh, Joanne Gudario. I was with Cornell for 40 years in the area of, I ended up the last 10 years in sports turf management, with the community and school feels as a priority, and safety, and using resources to make those fields functional and safe. but working with Nina, I arranged some of our visits and our communications with the different communities that are working with us. And I get so much joy out of seeing the commitment from these folks that are so happy to be part of this study as we go across the state and taking the data. And I am fading, so I want to just say that here's what was awesome for me: the passion, the commitment, of the science, the scientists blending the educational opportunities for your students, and to care so much about your environment. And that, I think that was awesome. And the fact that the cooperator, the Greenwood, to be so generous and giving us a living laboratory. I just think this was just, it's just awesome to be part of it. And I know we appreciate it. So I just had to say that, and I commend you all for doing your work. And I think the galls are cute. Okay, let's leave it like that. They're cute. And I do like lepidopters, but I am a grass person, but turned into a quercus. No, I'm, oaks. And I've just been,
We're sorry for disparaging turf earlier. We should have been kinder to the turf.
Oh, I always smirk at that because sometimes I do some talks about the ecological impact of turf grass, but like all the plants we have in our life, it's the mismanagement that gets us all in trouble. So anyway, it just was very uplifting to be part of this, to listen. And I know we'll see you again because we'll be back. And I'm sorry I have to leave, but thank you for everything, for all of you folks working hard and the generosity of Greenwood.
Thank you, Joanne.
All right. Take care.
All right, bye.
All right, bye.
And then one of the last questions to bring in, galls, particularly from oak trees, can be used to produce a wood stain, specifically dark black stain, due to the high concentration of tannins. More of a comment, less of a question.
And you can also use it to make ink. Oak gall ink was a thing, especially in Europe. And some really old medieval manuscripts are all written in oak gall ink. It's from particular galls. I don't know if they're the same ones that we have on the trees in Greenwood, but yeah, galls exist all over. They're not just here. And oak galls are really cool, obvious things, and people have been using them to do things like make ink or make wood stains because of the tannin concentration. So yeah, super cool.
Yeah, I think it's additionally, just to kind of the land use aspect, it's really worth noting where a given tree is growing, right? Is a tree growing right next to a road or a sidewalk? And right outside of Greenwood, it has all these major heat island impacts that Nina mentioned that are more severe and more concrete, which increases the alkalinity. So whether or not they're natives that can handle those conditions, I don't know as well. But a lot is kind of worth noting on where you can and cannot plant a tree. And a lot of our work, what we're suggesting is if you can plant a tree and it's a reasonably hospitable environment, which much of the suburbs are and a lot of parklands are, and some sidewalks, not all, for planting things, that a lot of these native trees really can be these wonderful nurseries, as Sarah put it, for bringing back wildlife into areas where they've largely been lost in the urban and suburban habitats.
Yeah, thank you, Michael. Yeah, for me, this just emphasizes the wondrous complexity of life that we are not real experts of yet, because questions still keep popping up. And I also really like that it really challenges old connotations and old perspectives, i.e. feeding insects are bad, so because not all it might depend on the insect.
Yeah, there was a fun article, if people want to read a little bit about gulls, in the New York Times. I'm not sure if it was Margaret Roach, about hackberry gulls, which are really abundant. You can see them in abundance in Greenwood or any other place that hackberries grow. But it was talking about how a lot of people in the South, where these grow more commonly, call them trash trees, but really about how much migratory birds use them and other species. So even sometimes it does make a tree that's not so attractive. it can also bring a lot of stuff in with that.
Yeah, such a rude name for that tree. Hackberries are one of my favorites. We actually have a state champion hackberry at Greenwood. That means it's the largest of its species in the state of New York. Come check it out. It's huge and it's beautiful and it is not trash. I love it. But to speak to that, that really changes attitudes and preferences. And, as land stewards and people, like those impact the choices that we make in terms of design and planting. Yeah. So just to give a critical thought to why we choose things and like things. All right. I think those are all the questions and thoughts for me. Emily, Michael, Nina, anything from you?
No, other than it's just been a joy to be here tonight, and it's so delightful to be a part of Greenwood and the Greenwood community and getting to spend time out in the cemetery, like Adana was saying, like Liam was saying, it's one of the highlights. Even when it's a million degrees hot in the summer, it's just amazing to be spending time out listening to the trees, out listening to the breeze, and enjoying the cemetery.
It's still a lot cooler there in the summer than on the street.
Yes.
Yes, thank you, thank you, Matthew Wills. Awesome. And I just want to do a little plug in. Check out the Greenwood event calendar. There are tons of birding tours led by our resident birder, Rob Jett, where you'll be able to see these birds in action, eat these insects in action that are feeding off some of these trees. And amongst them, there are also other walking tours that focus more on the smaller plants, the less charismatic flora, if you will. And then we actually also do tours. Matthew Wills, his name has been dropped now 4 times in this presentation, but he leads insect walks, and that's basically like a birding walk, but you're focusing in on insects. Well, anyway, thank you so much, and we'll probably see each other soon, shortly. All right, have a good night, guys.